All right, welcome everybody. Today, for those of you guys who are new to this, we have two talks. They're 20-minute talks, and we'll have 10 minutes for questions. You guys will be able to type your questions into the chat box, and then you'll be able to unmute yourself and ask the questions. And we've changed the format from when me and John were running it earlier. We only had students and postdocs giving talks, but now we've expanded it to junior faculty. And so this week we have our first set of 2 junior faculty. So our first talk today is from Angeline Lyon. She's an assistant professor at Purdue University. Angeline did her doctoral research at the University of Texas at Austin with David Hoffman, where she worked on biophysical studies of the RNA recognition by Bruno proteins. She then moved to University of Michigan, Ann Arbor to John Tesmer's lab, where she did some really amazing work, structural studies of PLC enzymes. And this is where I started to notice her work and followed it in great detail. She moved relatively recently to Purdue in 2014, and she's been very successful there. She's won a number of young investigator grants from the American Cancer Society, as well as the American Heart Association, and she's now funded by the NIH. And she's published a lot of very nice papers, and one of those papers is one that you'll hear about today. So with that, I'll hand it over to Angeline to give her talk.
Well, thank you so much for the introduction, Mike. It's a pleasure to be here. And thank you to you and John for organizing this. It's been really fun to watch this seminar series evolve from its early days back in the spring. So today I'll be telling you about one of the stories that we've been working on recently in my lab, and our effort to understand how phospholipase C enzymes are regulated. And in particular, I'll be telling you about PLC epsilon today. So first of all, just as a reminder, PLC enzymes are large multi-domain proteins that are typically present in the cytoplasm of resting cells, but they're translocated to cell membranes in response to different receptor stimulations. And there, they have canonically thought to hydrolyze the intermembrane component PIP2 into the second messenger's diacylglycerol and inositol triphosphate. So IP3 is a freely diffusible small molecule. It binds to receptors and promotes the release of calcium from intracellular stores. And this calcium, together with the diacylglycerol in the membrane, goes on to activate protein kinase C. So this places PLC enzymes upstream of a number of different cellular processes, including things like cell proliferation, differentiation, and survival. Now, there's actually 6 subfamilies of PLCs that can all hydrolyze PIP2. And shown here is the domain architecture for all 6 of them. And so there's a lot of information on this slide, but what I hope you'll take away from it is that overall, these colors tend to repeat, and each one of these colors corresponds to a conserved domain. So almost all PLCs have an N-terminal plextrin homology domain, and then all of them have 4 tandem EF hand repeats, a catalytic TIM-barrel domain that's split into halves by a regulatory XY linker, or in some cases, like in PLC gamma, by intervening domains, and then a C2 domain. And for a long time, the view has been that from the PH domain through the C2 domain, or depending on the subfamily, from the EF hands through the C2 domain, this region is the minimal functional unit of a PLC. So that's enough to give you some amount of lipase activity. But to get the subfamily specific regulation, you need to have additional either domains or insertions present. And so these are often at the C terminal of the protein, or in the case of PLC epsilon, at both the N and C terminal. And then again, there are some regulatory insertions that are commonly seen within the TIM-barrel domain. So today I'm just going to tell you about one of these subfamilies, PLC epsilon. And our interest in this enzyme is largely from the fact that it's regulated by G proteins and it's required for normal cardiovascular function. It's also been shown to contribute to both cancer and inflammatory pathways as well. And when in the next slide, when I tell you a little bit about its regulation, you'll start to see how it can do so many different things. So PLC epsilon has been best characterized in the cardiovascular system, where it can be regulated by G protein coupled receptors, including things like the beta adrenergic receptors or thrombin receptors, as well as through receptor tyrosine kinases. And in particular, this is growth factor receptors. Stimulation of GPCRs or RTKs activates G proteins, and then these G proteins can bind directly to PLC epsilon and translocate it from the cytoplasm to different cellular membranes. Now, depending on the pathway that is involved, PLC epsilon can be translocated to the plasma membrane, where PIP2 is the primary substrate, or it's also been shown to be translocated to the perinuclear membrane, and there PI4P is the predominant substrate. And so in both cases, you get signaling through diacylglycerol. Now, on the plus side, PLC epsilon is required for maximum cardiac contractility. And that's shown here at the bottom, where we're looking at calcium oscillations and the presence or absence of PLC epsilon. And so if you stimulate these cells with agonist, you can, and ultimately activate PLC epsilon, you can see that when the lipase is present, you have a robust increase in the amplitude of these calcium oscillations. But in the absence of the lipase, you see a blunted response. So your heart is still functional, but it's not up to its maximum capabilities. However, chronic activation of PLC epsilon, in particular through the beta adrenergic receptor pathway or through overexpression, can lead to cardiac hypertrophy and heart failure. And so understanding how this enzyme is activated at different places at different times by different G proteins is really important, and being able to distinguish between the pathways important for maximum cardiac contractility, but without that tipping into negative consequences. So PLC epsilon, as I mentioned, it's a big multi-domain protein. It's cytoplasmic, and it shares the same conserved core as many of the other PLCs. So again, the pH domain through the C2 domain, this element or this region is thought to be required for lipase activity. But PLC epsilon also has a number of unique regulatory domains that allow it to do and respond to all of these different pathways. So at its N-terminus, it has a guanine nucleotide exchange factor domain, the CDC25 domain, that actually recognizes inactivated RAP1A, the small GTPase, and then activates it by catalyzing exchange. And at its C-terminus, it has two RAS association domains, RA1 and RA2, that have evolved different functions. And so in addition to contributing to things like G protein activation, these regulatory domains have also been proposed to be important for subcellular localization. So how do you get from the plasma membrane or stay at the perinuclear membrane? Sorry about that. How do you get to the right membrane and stay there? As well as contributing to autoinhibition. And so efforts to understand how all of these pieces in PLC epsilon work together to give you this coordinated lipid hydrolysis output have been really challenging. And in large part, that's because of the size and the complexity of the protein itself. So it's over 220 kilodaltons. And so far, we actually still haven't come up with a way to express and purify enough of the full-length enzyme to do any sort of meaningful in vitro biochemical study, let alone structural studies. And so when we started working on this, we took a page from my postdoc basically and asking what's the minimal piece of a PLC that we can express and start to work out how these regulatory domains contribute from there. And so with PLC epsilon, the largest fragment we've been able to characterize is this PHC variant. So it starts at this N-terminal plextrin homology domain and contains the 4 conserved core domains. And then we also have the 2 C-terminal RAS association domains. So it's not a perfect system, but this does have robust catalytic activity, and we can get enough of it to do things like small angle X-ray scattering and electron microscopy. And from there, we can start to ask, well, what happens if we get rid of one or both RA domains, for example? Or if we make N and C terminal truncations. So it turns out that work from our lab as well as John Sondek's showed that you don't need the pH domain in PLC epsilon or the first 2 EF hands. And so by taking this approach and looking,
I think we lost Angeline, but hopefully she'll be back soon. So let's just give us a few moments. Okay, so I apologize. Unfortunately, this is internet connections with Zoom. But we'll go to Tiago, and then we'll finish with Angeline's talk when she gets back. So it's my great pleasure to introduce Tiago Oliveira. He is an MD PhD. So he's a practicing neuroradiologist at the University of Minho in Portugal. He did his MD PhD program at the University of Minho and Columbia University. At Columbia, he did his PhD work under Gilbert DiPaolo's lab, where he studied the role of lipid signaling in Alzheimer's disease. And he showed that ablating the enzyme Phospholipase D1 was protective in different Alzheimer's disease models. He since started as an assistant professor in 2011, where he's continued to study Alzheimer's disease as well as other mood disorders. So with that, I'll give it over to Tiago, and he can present to you his exciting work.
Yeah, thanks for the introduction, Mike. And hopefully, this will go smoothly. It's always a surprise with Zoom. And today I'll be talking about the role of phospholipase D1 in hippocampal functioning. And first of all, I would like to give credit to the two main contributors for this work, Luisa Santamaria. She was the main driver of the work, and she's also an MD, and she's finishing up her PhD at Minho under my supervision, and she's currently applying for residency. And also Isabel Castanho, she was a member also of my lab a few years ago, and she's now currently a postdoc at the University of Exeter in England. So one of the main topics that we are interested in my lab is the study of Alzheimer's disease. And Alzheimer's disease is characterized by memory deficits, mood alterations, and also neurodegeneration with atrophy in different regions of the brain. But one of the features in neurodegenerative diseases is that it affects preferentially certain regions. So for instance, in Alzheimer's disease, it affects preferentially, among other regions, the hippocampus. Then that's also one of the other reasons why we are interested in studying the function of this brain region. The hippocampus is localized in the temporal cortex. And to study the hippocampus, not only we do human studies, but we also make use of rodent models. And here I'm representing a representation of a coronal section of a mouse hippocampus. And here we have its different subfields, the data gyrus, C1, C2, and C3. And this organization, this coronal organization, is somewhat maintained along the longitudinal hippocampal axis. And various research groups have been studying this organization of the hippocampus. And in a somewhat simplistic way, they have segregated the most dorsal portion of the hippocampus, this dorsal pole, from the ventral pole of the hippocampus. And even though these poles and this axis is organized along a gradient of this longitudinal axis, in a simplistic way, some functions have been attributed to each pole. For instance, the dorsal hippocampus has been more implicated in specific dimensions of spatial memory, and the ventral hippocampus has been implicated mainly in some emotional associated behaviors. So another topic that we are interested in my lab is the study of lipids. And since lipids are one of the main constituents of the brain, one of the strategies that we use is to study certain regions of the brain. We extract the lipids from those regions, and then we perform lipidomic analysis. And we can, with this approach, we can have an idea of physiological and pathological signatures that could be giving us some hint in how the brain functions in disease and in the physiology. And that was more or less the strategy that we used to understand this question of what are the lipidomic signatures, a characteristic of this longitudinal hippocampal axis. And to do that, we divided the hippocampus in three parts, in dorsal, intermediate, and ventral portions. And here I'm going to show our lipidomic analysis of the dorsal and ventral hippocampal signatures. So here I'm showing in black the dorsal hippocampus, and the ventral hippocampus is here in red, which is the normalization that we did here is the ventral hippocampus relative to the values of the dorsal hippocampus. And overall, some of the things that we see was that in the ventral versus the dorsal hippocampus, there was a differential single lipid regulation. We also saw evidence for a differential glycerophospholipid regulation. And at the pathway level, we saw that the dorsal hippocampus had higher levels of phosphatidic acid and lower levels of phosphatidylcholine. And there is an enzyme that catalyzes this reaction, which is phospholipase D. And phospholipase D is one of the major sources of phosphatidic acid. And phosphatidic acid is at the center of a very complex lipid signaling network. In mammals, there are two main canonical phospholipase D isoenzymes, PLD1 and PLD2, that differ in terms of their structure by the presence of a loop region in PLD1. And in terms of the reactions that they catalyze, in the presence of water, they produce phosphatidic acid. However, phospholipase D has this very characteristic property that in the presence of a primary alcohol, such as ethanol, it preferentially uses it to produce, in this case, phosphatidylethanol. And this has been used to measure PLD activity in vivo by measuring phosphatidylethanol, and we can dissociate from other sources of phosphatidic acid. And also researchers have used in the past this property to inhibit PLD activity. Nowadays, we have the ability of using specific inhibitors, and we have improved, in that sense, these studies. In terms of its localization, PLD1 is more in the Golgi, secretory granules, endosomes, and autophagosomes, and PLD2 is more in endosomes and in the vicinity of the plasma membrane. So to actually study the role of phospholipase D1 and D2 in the brain, we made use of knockout models for each isoenzyme. And we also crossed these two isoenzymes to produce the PLD double knockout animals. And we use here, just to confirm these knockouts, an antibody for the C-terminus that can recognize both PLD1 and PLD2. So here we could confirm that we are working with the right models. We also did some functional studies to measure PLD activity in vivo. And to do this, we use that same property that I described earlier for the higher affinity for primary alcohols that phospholipase D has. And we made use of a method that I described and I developed earlier on during my PhD, where we can inject ethanol in mice. And 1 hour afterwards, we can, in this case, dissect the brain. I dissected the forebrain and measure the levels of phosphatidylethanol as a surrogate for phospholipase D activity in a given brain region. So we used this in the past to show that the activity of phospholipase D was increased in an Alzheimer's disease mouse model. And here we confirm that the activity of total PLD activity levels in PLD1 and PLD2 were going down. We can see here the levels of phosphatidylethanol in black comparing with the levels of wild type animals. And we could also confirm that there was basically residual or almost no activity in double knockout animals. And we also had as a control group non-injected animals. So overall, this suggests that PLD1 and PLD2 are in the brain at least, the two only sources of canonical PLD activity. And if you actually look into this and if you sum up the reduction of PLD1 and PLD2, you see that it doesn't sum up to 100%. So it also just indicates that there is some level of compensation going on in PLD1 and PLD2 knockout animals. So we also looked into more specifically at the species of phosphatidylethanol. And we could see that as more or less expected, the majority of the species of PLD double knockout and non-injected animals were decreased. In PLD1 knockout animals, there was also a big array of phosphatidylethanol species that were going down. And then in PLD2 knockout animals, we could see that phosphatidylethanol species that had a shorter chain length, fatty acid chain length, they actually also had decreased levels. But somewhat surprisingly, there were some species that had fatty acid chain length that was longer. They were actually increased, which further supports that there are some compensatory mechanisms. In this case, it further actually supports that it might be some kind of overcompensation going on. Then we went to that question that was suggested by one of our earlier studies, that the phospholipase D pathway could be involved in the regulation of the longitudinal hippocampal axis. And to do that, we did a lipidomic analysis of dorsal and ventral hippocampus in PLD1 knockout animals and PLD2 knockout animals. And the values that I'm showing here in these heat maps are relative to wild type littermates. And the first thing that we can see here is that in PLD2 knockout animals, we didn't observe any significant change at the level of the different lipid classes that we looked into. Then in PLD1 knockout animals, we could see that the levels of phosphatidic acid was actually going down both in the dorsal and ventral hippocampus. And looking into other lipid classes, there were also other changes going on. And we saw that, I would highlight the changes in the dorsal hippocampus in different lysolipids. Then we also looked into, at the species level, in this case of phosphatidic acid, we could see that in PLD1 knockout animals, there were multiple number of species going down, both in the dorsal and ventral hippocampus. And then in PLD2, we saw only decreased levels of phosphatidic acid species in the ventral hippocampus. And again, somewhat surprisingly, there was at least an increase in one species in the dorsal hippocampus that was also with a fatty acid chain length that was longer. So this could also further suggest that there is some kind of overcompensation, as we saw before. To understand if this compensation was dependent on the protein levels of each enzyme counterpart, we saw that in PLD1 knockout animals, the levels of PLD2 were normal. And in PLD2 knockout animals, the levels of PLD1 were normal. But what was interesting when we look into this was also that comparing dorsal hippocampus with ventral hippocampus, that the levels of PLD1 in the dorsal hippocampus were higher comparing with the ventral. So this also aligns more or less well with a higher effect of PLD1 ablation in the dorsal hippocampus. So from now on, because of this predominant change in phosphatidic acid levels in PLD1 knockout animals, we focus more in the characterization in more detail of PLD1 knockout animals. And one of the first things that we did was to perform an array of different behavioral analyses that relied at least partially in the hippocampus. So I'm not going through all the behaviors that we did. It was a lot of work, but I'll describe more where we actually found some significant changes. I should say at least that we didn't find alterations in PLD1 knockout animals at the level of exploration in the open field task, no changes in anxiety-like behavior in elevated plasmase, no changes in fear memory or spatial memory in using the Morris-Water maze paradigm, and no changes in sociability. So concerning the tasks that we actually observed changes, in this task that is called novel object recognition, we place the mouse in a box where he has the possibility to interact with two equal objects. Then we take the mouse out of the box, And then after a while, we expose the mouse again in that box. But in this case, we change one of the objects. And this case is a new object while the other is the same. Mice are naturally curious, so they tend to spend some time preferentially exploring the novelty. And this is actually what we see here in the wild type, that animals are exploring more the novel object. In PLD1 knockout animals, even though there is a tendency for this higher exploration, what is actually more apparent is that these animals are exploring more time the familiar object. So when we calculate the index of discrimination of this novel and familiar object, we actually see there is a change, even though it's subtle in this paradigm. Then, also inspired by these changes in exploration, we tested other exploration behaviors that in this case are reliant in social circuits. And in this case, the paradigm that we did was to place one animal in a cage after different habituation steps. And in this situation, the mouse has the opportunity to interact with a litter mate that he knows very well, and with a novel animal that we name here as a stranger. And again, mice are curious, so they have the tendency to interact more with a novel animal. And we can see here in wild type that is actually what happens. Also in the PLD1 knockout animals, that also happens. We didn't see in the discrimination score any major change. But when we actually calculate the total time of exploration, in total, PLD1 knockout animals were less social in this task. And I should highlight that in this specific task, this is actually specifically dependent on the hippocampus. So then we wanted to understand what was going on in the hippocampus more specifically, if we could more or less explain what we are actually seeing in these behaviors. So we performed a Golgi-Cox impregnation preparation to allow us to perform 3D neuronal reconstructions. And this is more or less the kind of reconstructions that we can do from, in this case, pyramidal cells in CA1 and CA3. And we also did granule cells in the data gyrus. And we did this both for dorsal and ventral hippocampus. So that's more or less the summary of all the results that we see. And just, it's a lot of data, but comparing just the wild type with the knockouts, the only difference directly that we saw in terms of this comparison was a decreased dendritic length in dorsal hippocampal apical dendrites in the knockouts. But there was another feature that was interesting. We saw that in wild type, in CA1 and CA3, that there was a differentiation between the parameters such as dendritic length, the complexity of the arborization as the number of nodes, or in this case, the spine density, between dorsal and ventral. However, this differentiation was not observed in many situations in the PLD1 knockout animals that we are looking in terms of these parameters, which suggests that in PLD1 knockout animals, there is a disruption of this differentiation between dorsal and ventral hippocampus. Then we tried to see if there were any parameters that were altered at the level of electrophysiology. And the two parameters that we tested out were long-term potentiation, LTP, and long-term depression, LTD, which are related with forms of plasticity that are associated with forms of learning and memory. And to do this, we stimulate in the Schaffer collaterals, and then we record in CA1. So if we give a high frequency stimulation, we have LTP, and if we give a low frequency stimulation, we have LTD. And we did this both in slices for dorsal and ventral hippocampus. And then what we actually saw was that concerning LTP, we didn't see any changes either in the dorsal or ventral hippocampus upon PLD1 ablation. But then concerning LTD, while we didn't see any changes in the ventral hippocampus, we saw that PLD1 ablation led to a reduction in the induction of long-term depression specifically in dorsal hippocampus. it was interesting that while our paper was under revision, another paper had come out that showed exactly, or more or less the same, also in PLD1 knockout animals, a reduction in LTD induction. However, in this case, it was in another region, in the prefrontal cortex. And also, there was a previous publication that showed that long-term depression was associated and partially necessary for novel object exploration. So this actually connects both our behavior and our electrophysiology findings. And finally, we also performed Western blot analysis to measure if there were alterations at the level of synaptic proteins. And we tested different predominantly presynaptic or postsynaptic proteins. And from all the proteins that we look into, we found two alterations comparing the reduction of PLD1 levels that were in SNAP25 and in GLUN2A. And from these two, actually the most intriguing, the one that was most connected with the literature, was in SNAP25. More or less trying to explain and connecting this with the literature, an ortholog of SNAP25 in yeast was actually previously shown to bind to phosphatidic acid. And this is SPO20 that has been used by many research groups as a phosphatidic acid probe. Also, the exposure to a peptide that inhibits SNAP25 was shown to reduce LTD induction. And finally, SNAP25 heterozygote animals had deficits in object recognition and in different social behavioral paradigms. So in summary, we show that hippocampus can be subdivided in distinct portions along its longitudinal axis, also based in its lipid composition, that PLD1 is the major source of phosphatidic acid, at least comparing with PLD2. PLD1 knockout animals had deficits in novel object recognition and in social exploration, and also had deficits specifically in the dorsal hippocampus in long-term depression. And then we also saw, partially explaining some of these changes, a decrease in SNAP25 and in GLUN2A. And this overall highlights that PLD1 and the PLD pathway could be related with different psychiatric disorders, and aligns also with some publications from other people. And we are definitely looking into this with more detail. And I just want to thank the people that contribute to the work, my current students, Luisa and Rafaela, and previous student André, Turcato, help with social behaviors, Fernando and Vitor, who are major contributions, my past students, people at Columbia, most of them are not at Columbia anymore, and Marcus also for his early collaborations, and I'll be happy to answer questions. Thanks.
Thank you, Tiago. That was an excellent talk, and thanks everyone for staying on. Angeline's back with us, so she'll be able to finish up her talk after Tiago answers your questions. So our first question is from Joyce Lyon. Do you want to unmute yourself and ask the question?
Oh, hi. I want to see, Angelina, that the screenshot that when suddenly cut off, because I was having some, I was trying to write down a few notes. So I want her, if she is okay, to show that screen again, then explain a little bit more deeper.
Okay, so we're taking questions for Tiago right now, but so Angeline, will she'll finish her talk. She's back on. So then our next question is from Nicola Vitali. You want to unmute yourself?
Yeah, great work, Tiago. Do you have an explanation for the more dramatic effect of the PLD1 knockout on PA levels, while in vitro experiments have indicated that actually PLD2 has a much higher basal PLD activity?
Can you repeat, Nicolas, again the question? Sorry.
Do you have an explanation for the more dramatic effect of the PLD1 knockout on PA level? Because in vitro experiments have shown that PLD2 has a much higher basal activity.
Yeah. So first of all, this is a key question, and just for the sake of time, I didn't also show some of your work, and you actually had a recent work that at least to confirming what we saw, that in adrenal glands, in PLD1 knockout animals, also the measured PA levels that go down is actually in PLD1 knockout animals. Yeah. Also from our previous work in the Neuroscience paper in 2010, we saw that upon the ablation of PLD2, also total levels of PA were not going down. So it actually seems that if anything, it's about specific phosphatidic acid species. Mm-hmm. That said, trying to connect with in vitro experiments, I think some of the things that we also highlight here is that cell type specific, probably either PLD1 and PLD2 would give a more predominant effect. Also, I'm here showing the brain, but this is similar to any kind of lesion or organ in the body. PLD1 or PLD2 could actually have a predominant role in one or the other. And there is also work from other people, also in cell lines, from instance from Jeremy Baskin's lab, where if you actually activate either more PLD1 or PLD2, you actually recruit, depending on the receptor that you're activating, either one or the other. So it actually depends a lot on the conditions. And I don't think we can say PLD1 and PLD2, they are the major ones. The data that we have from the animal models is PLD1 that is contributing the most for PA levels. But I think we need to always keep in mind the context and the situations that we're looking at, and especially the on-demand effects of PLD1 and PLD2, because a lot of their effects, it's upon a stimulation in a given context. So, you never know if a cell line is giving that stimulatory constant effect. So we have to keep that in mind.
Okay, thank you.
Thanks.
Binks, did you want to ask your question?
So you sort of touched on this, but it's, I'm wondering whether you think the effects are in the neurons themselves or in glial cells, and if there's anything known about the levels of PLDs in the different cell types.
Yeah, so that actually is still more or less, there is no clear answer for that. The experiments that we did were a lot in tissue, so we cannot dissociate the cell type specific experiments. One way to actually go about that would be to do like sorting of one type or the other and do lipidomic experiments again to actually do this. I would love to do those experiments. I really want to do those experiments. We haven't done, but I think that's one of the ways to go. Another way is to do cell-specific cultures. We've done in the past with neurons, where we show that PLD knockout neurons, for instance, in that case, have decreased total PLD activity. But I think this needs always to be redone. I should also say that the levels of PLD1 and PLD2, they are not so high in neurons, at least based on Ben Barry's database that was published a few years ago. So we also need to keep in mind that PLD1 and PLD2 could have a very important role in other cells in the brain that are not neurons. So it's not a question that has a straight answer as of now, but there is evidence that it has a role in neurons, and I think this should be appraised in more detailed studies. But yeah, I think this is more or less the evidence that we have out there.
So in principle, you could do cell type specific knockouts as well.
Oh, and yes, so you can also do that. Yeah, of course. On top of doing the sorting experiments to get the cell-specific measurements in terms of either the lipidome or even PLD activity levels with measuring phosphatidylethanol, you can also do cell-specific knockouts. Yeah, absolutely.
Dan, did you want to ask your question? This might have to be the last question. I apologize, just for interest of time. Dan, you're muted.
Sorry, this will be very quick because some of my questions were already asked and answered. Tiago, do you know if in your ethanol experiments, if the distribution of ethanol reaches equivalent parts of the brain, that there's not a problem with ethanol being unevenly distribution in the brain regions that you're looking at, which may explain some of the phosphoethanolamine data? Phosphoethanol data.
Yeah. So one thing I should say is that we looked exactly in the same region for all the knockouts. So we would control at least for that availability in different regions. That said, I I don't know exactly the answer to that. I would expect that alcohol is very diffusible and that it would reach more or less the same different parts of the brain. However, I think that needs to be looked into. And also another thing that needs to be looked into is that, and I don't think this has been done carefully, which is to measure the PLD activity in different parts of the brain. So not only if the ethanol reaches there, but if the activity is actually the same, and this relies on either PLD1 or PLD2. So I think it's these two questions. I would say that ethanol more or less reaches the brain, but I think the thing that would be really interesting to test would be if PLD activity is different in different parts of the brain, and if PLD1 or PLD2 contribute differently to this. I would say yes, but of course experiments need to be done to do this.
Well, thank you, Tiago. That
Thanks.
was a wonderful talk. And I apologize to the questions we couldn't get to. But maybe you can answer those in the chat box.
Yeah, and if you have questions, you saw my email, please email me, or on Twitter, and I'll be glad to discuss this further. And yeah, thanks. I'll try to answer in the chat box, the other ones.
Okay, so we are welcoming back Angeline. She's going to continue her presentation. I guess she can explain, but I guess there was a building-wide internet outage, so please forgive us.
All right, thank you all so much for bearing with me. I think that was bound to happen at some point, so anyone who's scheduled to speak next, you can not stress over what happens in this situation. So I think this is where I left off in telling you about our efforts to use a domain deletion strategy to basically map how different parts of PLC epsilon within this big PHC variant can actually talk to one another. And so for this, we can look at two things relatively quickly. We can look at the thermal stability of our PLC epsilon variants. So in black is our PHC variant. And in this assay, we're looking at the thermal stability. So this is differential scanning fluorimetry. And we take our purified proteins, mix them with a hydrophobic dye. And as the protein denatures, the dye binds hydrophobic spots and then fluoresces. And so the midpoint of this denaturation curve corresponds to the melting temperature. And so for our PHC variant, it has a TM of about 51 degrees. And so by taking advantage of all of these domain deletions, we can ask, well, what happens if we get rid of, for example, the RA1 domain or both RA domains? And you can see that the thermal stability actually decreases. And so what that tells us is that in this case, the RA1 domain is actually needed for the structural integrity of the rest of the enzyme. On the flip side, if we get rid of the pH domain and first two EF hands, which as I mentioned, we've shown as well as on Sondek's group that these are dispensable for activity, you can see that we can actually increase the thermal stability. And so this would mean that these regions, so again, the pH domain and EF1 and 2, are not important for structural integrity. And so whether that means they're flexibly connected or just transiently interacting with other regions in the lipase is something that we can start to look at further. Now, thermal stability is great, but ultimately we want to see how these elements contribute to catalytic activity. And so using either cell-based assays or liposome-based activity assays, we can again ask what happens. If we have the full length, our PHC variant, and we get rid of the RA1 domain, you can see that there's about a 20-fold decrease in activity. And so that makes sense given how much the enzyme was destabilized by deleting that domain. In other cases, getting rid of a domain doesn't really do that much to activity. So for example, getting rid of the RA2 domain or even the pH domain, first two EF hands and RA2 domain doesn't really do too much to activity. So putting all these pieces together, along with a lot of solution structure work and lower resolution techniques, so things like small angle X-ray scattering and negative stain electron microscopy, we can actually start to put together and map out on the PLC epsilon domain diagram, what parts do we actually have to have for catalytic activity, what parts are nice to have, and with the ultimate goal of figuring out at the atomic level, how does PLC epsilon actually work? So we know that the N-terminus, in particular, the region that contains the CDC25 domain, has really not been characterized, other than the fact that there is some GEF activity here. We know that the pH domain and first two EF hands are, again, as I mentioned, are dispensable. But we also showed using SACs and negative stain EM that these regions are also conformationally dynamic in PLC epsilon, as well as PLC beta. So for a high resolution crystal structure, this is sort of the kiss of death in terms of being able to do that. And even with all the advances in single particle electron microscopy, going after conformationally heterogeneous samples is still really technically difficult. Now, in terms of the rest of PLC epsilon, the region from the EF3-4, so the second half of the EF hands, through the C2 domain, this part is absolutely required in every PLC enzyme. And so this was, we didn't do anything there. But at the C terminus, we can again make some judgment calls. So the RA1 domain is required for activity and stability. RA2 is actually flexibly connected, as was shown by Matilda Catan's group. And so we decided to remove that domain as well, again, with the goal of coming up with something that would be conformationally homogeneous, as well as have catalytic activity. And so that left us with this EF3 RA1 variant. So we sacrificed about twofold basal activity with respect to our PHC variant, but this is a very stable protein and it's expressed very well in insect cells. Now, we had done so many other structural determination techniques and ended up being limited in part by resolution and conformational dynamics. But one of the first things we wanted to do was to check and see whether this EF3 RA1 fragment really did behave the way that we had hoped it would. And so using both SACs shown on top as well as negative stain EM on the bottom, you can see that from our Gugnier plots and our pair distance distribution functions, that as well as our SACs envelopes and EM reconstructions, even though they're very low res, this is a pretty compact structure. So there's not beads on a string or anything like that. And moreover, our studies indicate that it's monomeric and monodisperse. So this is a great target to try and go forward with doing high resolution structural techniques with. And so we were able to solve the first crystal structure of PLC epsilon, and that's shown here in this 2.7 angstrom structure. And then the little crystal up here is actually the one that was used to collect this data set. So within the ribbon diagram, again, all PLC shared from the EF3-4 hands, the catalytic Tim barrel domain, and the C2 domain. So this region of the molecule actually looks very similar to what we see in PLC delta, beta, and gamma, which makes sense because this is really the catalytic machinery that's holding the protein together. Now, that being said, there are some really important structural features that I want to highlight. So first off, PLC epsilon is auto-inhibited by this XY linker region. And I'll talk more about this in a moment, but one thing that was really unusual in this structure is that we have two very well-ordered elements. In most structures, this linker region is disordered. But PLC epsilon has a very well-ordered amphipathic helix on one side, and then a beta hairpin on the other. And so since we don't know how this helix or this linker regulates basal activity, having these structural elements can give us a foothold into exploring this further. Now, the most obvious thing that sets apart this particular crystal structure is the presence of the RA1 domain in PLC epsilon. And so the RA1 domain is connected to the C2 domain by the C2 RA1 linker. So this is a relatively well-ordered linker. It's pretty short, but it packs against the Tim barrel and the C2 domains. And one thing that was really gratifying about seeing this structure is that it provided a structural explanation for the biochemistry that we'd done previously. And that deleting this linker and the RA1 domain, so effectively truncating our protein immediately after the C2 domain, that really killed activity and stability. So about a 20-fold decrease in stability and a 6 to 7 degree decrease in stability. And so this interface varies over 1,600 angstrom squared surface area, which is quite extensive. So removing this really does significantly impair the ability of the enzyme to adopt the right conformation to carry out that PIP2 hydrolysis function. And so with that in mind, though, one of the caveats is always, in a crystal structure, you're at such incredibly high protein concentrations that it's not uncommon to start to see things just sort of packed together. And so to make sure that these interfaces that we observed were actually forming in solution and had some sort of physiological relevance, we again went through and did some site-directed mutagenesis. So here's the overall crystal structure again. And I'm just going to focus on this linker and RA1 domain first. So these are just close-ups of how that linker region packs against the C2 domain, the timbarrel domain, as well as the loop that connects the two of them. And then this extensive interface is formed by the RA1 domain. So in order to look at this in vitro, we made individual point mutations in the background of our PHC protein. And we asked simply whether one or more of these regions was particularly important for maintaining the overall structural integrity of the lipase. And so when we mutated the C2RA1 linker, whether it's binding site on the core or the linker itself, we could decrease stability by about 1 to 3 degrees. Whereas if we did the same mutagenesis strategy between the RA1 domain, the C2 domain, and EF34, we saw larger decreases in stability. So this all makes sense. So this interface really does form in solution, and it is important for structural stability. And we think that, at least in the case of PLC epsilon, that maybe we need to redefine what we think of as the minimal catalytic core to really reflect the fact that this RA1 domain is playing a very basic functional role here. So in PLC epsilon, that core may really be EF34 to the RA1 domain. Now, when we went and looked at basal activity, we did this both in vitro in our PHC background, as well as in cell-based assays where we can work with the full-length enzyme. And surprisingly, it was only when we mutated the C2RA1 linker that we actually saw changes in activity. And instead of being losing activity, we actually found that mutations in this linker are activating. And so this linker is, in fact, an autoinhibitory element that is packed against the catalytic core. And we saw that these mutations were sufficient to increase activity both in vitro and in vivo. And so in a few minutes, I'll tell you how we think that this linker region could actually be displaced in the context of PLC epsilon's activation in the cardiovascular system. Okay, so switching gears and going back to this XY linker. So it had been shown several years ago by John Sondek's group that if you delete the entire XY linker in either PLC epsilon or in other PLCs like beta and delta, you could dramatically increase basal activity. And so this linker is quite large in PLC epsilon, it's 110 amino acids. But you can see that when you remove it and look at cell-based activity, you see this massive increase. So somehow it is regulating activity. And the idea is that, well, perhaps they act as effectively lids over the active site. And then as PLC epsilon or your favorite PLC of interest interacts with the membrane, either by itself or in complex with a G protein, then you get interfacial activation. And maybe that is somehow pushing these linker regions out of the way. So to start investigating that in PLC epsilon, we again went and made sure that these elements weren't doing something really important in terms of structure. This alpha XY helix is actually really interesting. So it's mediating an important crystal contact within the crystal lattice, but in solution, it does not interact with the PLC epsilon core. So it's not forming any intramolecular contacts. And this helix is also amphipathic. So if you wanted to design a perfect helix to bind a membrane, you would have to look pretty hard to beat this one, as it has a beautiful mix of hydrophobic residues and basic residues on the other face. And so when we deleted either one of these elements, nothing happened in terms of basal activity. But surprisingly, deleting this alpha XY helix really decreased activity in cell-based assays and almost to background levels. And so even though that this helix is autoinhibitory, there's something special about whatever it is this alpha XY helix is doing. And because it's amphipathic, one thing that it may be doing is actually regulating membrane association. And so one of our projects right now is really investigating how this element is making such a big impact in terms of basal activity. So starting to put all of these pieces together with what's known in the literature, as well as the cell biology studies that have been done, we think that we can start to propose a model for autoinhibition, as well as activation downstream of beta adrenergic receptors. So in the heart, PLC epsilon is present in the cytoplasm. We think I showed you that the EF34 hands through the RA1 domain that forms a very stable, compact structure. This is all you need for lipase activity. And in fact, this is actually auto-inhibited by both the XY linker as well as the C2RA1 linker. Now, what happens outside of this core is a little bit more difficult to say. We know that the pH domain and first two EF hands are conformationally dynamic, but that does not mean they're not transiently interacting, and they do contribute to basal activity positively. The RA2 domain is flexibly connected as well, and then the CDC25 domain, again, is not characterized yet. But when we stimulate cells with a agonist, like adrenaline, for example, a lot of different things happen that can culminate in RAP1A dependent activation. So RAP1A is going to do several different things to PLC epsilon. It's known that it binds directly to the C-terminal RA2 domain, and so this interaction is actually sufficient to localize PLC epsilon to the perinuclear membrane for PI4P hydrolysis. Now, because we're getting membrane localization, that's going to promote interfacial activation. So that's probably going to induce some rearrangements within the XY linker. And maybe this alpha XY helix is important for helping stabilize its contacts at the membrane. Now, the RA1 domain, I've only told you about its role as sort of what appears to be a scaffold for the rest of the core domains in PLC epsilon. But in fact, it can also interact with a scaffolding protein called muscle-specific A-kinase anchoring protein. And while we don't know the surface of the RA1 domain that makes this contact, given how tightly it's associated with the rest of the core, it's not unreasonable to expect that this MA-cap RA1 interaction may be sufficient to actually displace the C2RA1 linker. And so you would get allosteric activation as well, potentially by RAP1A binding, but also by binding to the local scaffold. And so clearly we have our work cut out for us in figuring out more of the details of this mechanism, and certainly what all of the regions in PLC epsilon are doing to contribute to this. But with all the timing issues in mind, I want to at least quickly acknowledge the people who've really driven this work. So much of this was done by two former students, Yvonne Rogema and Monita Singh. And our current projects are being carried out by Liz Garland-Kuntz, who's been a staff scientist through all of this, Dr. Isaac Fisher, Kaushik Muralidharan, and fantastic collaborators and graduate students. And we've been very generously supported by the AHA and NIH. So thank you so much for bearing with me. That was unexpected, but I would be happy to answer any questions if we have time.
Thank you, Angeline. That was fantastic. And you did a great job with your coming back afterwards. So,
Kind of you.
Yeah. So the chat is now open for questions. I had a question. So I'll start it off. I guess I'm curious about that XY linker because I know in the other PLC family members, it's thought to be an autoinhibition domain, and there's certain elements that sit above the active site. If I'm not mistaken, do you see those same elements in PLC epsilon?
So yes and no. So PLC epsilon, the N-terminal ordered, the alpha XY helix, that's not been seen in any of the other lipases to date. At the C-terminal ends, though, so PLC beta and delta both have a region that's at least, that's ordered and at least near the active site. And in one of the copies on our crystal structure, we saw sort of a run of amino acids over the active site and stabilized by that beta hairpin. But it was only in one of the subunits. And so we were initially really excited that, oh, maybe this is a shared mechanism of activation. We don't really see that though, when we delete the hairpin, for example, and the activity doesn't really change. That's not to say that there's not some commonalities though. So beta, delta, epsilon, within that XY linker, the only part that's really conserved across subfamilies is an acidic stretch. So there's 10 to 15 glutamic and aspartic acid residues. And those are really critical for this autoinhibition process. But where they're at moves within the linker. And so that's something that we're trying to figure out. So there's probably a shared overall mechanism, but the details are subfamily specific.
Very nice. Let's see, is there any other questions coming in? So I guess I'll ask another one. If someone wants to chime in. But I guess I'm wondering about why do you think when you delete those like the pH domains and the the EF1 and 2.
Mhm.
And the protein becomes more active, if I remember right, it becomes more active but less stable. Is there, do you think the enzyme, how do you explain that, I guess? I'm just kind of curious.
Yeah. So that's a really good question. So when we get rid of the pH domain and first two EF hams, we do see a slight increase in stability and a slight decrease in activity. So we've done a little bit of work trying to figure out if the PLC epsilon pH domain is contributing to activity by membrane localization. And so this has really not been looked at in PLC epsilon. You know, how does the protein get to the membrane and then stay there? So it's possible that the pH domain, you know, it's flexibly connected, but it's really maybe acting as more of a lipid sensor, like in PLC delta. And I don't have a definite answer for you, but it's like it's.
Can you hear me?
Oh, who is, I can hear you, yeah. Can we mute? Okay, well that answered my question. So there's a question from Anne Gershenson. You want to unmute yourself?
Hi, Angelina. Angeline, that was a lovely talk. I have a question actually about your thermal denaturations, because they mostly look two-state, and then there's one that looks like it's three-state, and I realize you might not have the structural information to know what's going on there.
Yes, so that's a really interesting question. So we do usually see more of a two-state behavior, but some of our variants look like they have a three-state model, as you said. We don't have the structural explanation for that yet, but given that some of these domains are, you know, at least transiently interacting or flexibly tethered, I think that might be accounting for at least part of it, where, you know, the individual domain unfolds or starts unfolding before the bulk of the protein does. But that's certainly something that we're continuing to follow up on. And it's a great question.
Thank you.
We had a prior question from Joyce. I don't know if Joyce is still here.
Yeah, I'm here.
Okay, did you want to ask your question?
No, I don't have further question.
Okay. Okay. Well, I want to thank everyone for attending then. And I want to congratulate our speakers, Tiago and Angeline, for fantastic talks. We will see you guys all next week for lipid signalings from flies to worms. And I hope you guys all enjoy the rest of your week.